Hello and welcome to the lecture series on Animal Kingdom. So today we are going to discuss the first major phylum of the animal kingdom, and that is Porifera, phylum Porifera. Okay, so let's start. Well, the phylum Porifera, or we can say the term Porifera, it's it's actually it is actually the combination of the two words. One is called the porous. So porous means opening, and fear. Fear means to bear. Okay, so this is actually the combination of the two words. These were recognized, these poriferans, these were recognized as animals by Alice. So Alice was a great biologist, so he recognized them as animals, or we can say he included them in the animal kingdom. The term porifera was given by Robert Grant. Okay, or Robert Grant. They are the most primitive animals, popularly called as sponges. So they are also called as sponges. Most of the scientists today hold of the view that the porifera is actually the old name, and now they are today they are called as sponges. Okay. Well, there is a separate branch that deals with their study. Okay, or we can say the branch that deals with the study of the sponges is called the parasology. It's called as parasology. Okay. Now, most of them are marine. Mostly are marine, and are sessile. Well, actually, what does it mean? Sessile means that they remain attached to the rocks. Attached to the rocks. Although few are freshwater animals. Although some of them are freshwater, just like the best example for the freshwater is spondula. Okay, so spondula is actually a freshwater sponge, or we can say the poriferan. So this is the only phylum in the animal kingdom without any nervous cell. Only phylum without any nervous cell. Any nerve cell. So there is no nerve cell. And they have already, I have discussed it with you that they have cellular level of organization. Okay, so these are the some we can say the few introductory lines that you should keep in mind. Now we are going to discuss very important few things here, and we call them as the important characteristic features, we can say. So let's study. Let me choose here separate color. So I will write down here important characteristics. So okay, important characteristics. Now, as I told you that they are marine and few of them are freshwater sponges, and their man attached to the rocks that they are sessile. Now, the first and the most important characteristic is that they are asymmetrical. They show asymmetry. While some are in their adult stage, we can say some show radial symmetry. Some, I will write down here, some. So some show radial symmetry. And they are pore-bearing animals. Pore-bearing animals. Pore-bearing animals, because their body contains numerous openings or pores. Numerous openings or pores. Okay, called as ostia. Well, the ostia is actually, this is a plural term, and its singular term is, sorry, this is a singular term. We call them as just one pore. We call it as ostium. Okay. And talking about their body wall, so let me choose a different color for this body wall. Talking about the body wall of the poriferans, the body wall of the poriferans consists of following layers. Now, our first is, we call it as pinacoderm. Pinacoderm. Well, it's also known as dermal layer. It's also known as dermal layer. And its outer cellular layer, you can see, I will also show you in a figure. It's also a outer layer, outer cellular layer, and it consists of two types of cells. Okay, it consists of two types of cells. One is called as gonacocytes, or we can see the gonacocytes. Sorry, wait a second. It's actually gonacocytes. Gonacocytes, and these pinacocytes, they are actually flattened. In their shape, they are flattened. Now, talking about another group of cells, another kind of cells, or we can say the second kind of cells, we call them as porocytes. Okay, and for your kind information, they are oval in shape. They are oval in shape. Well, what we learned that there is a layer, and that's the outer layer, and we also call that as the dermal layer. And its outer cellular layer, it consists of two kinds of cells. One is called the pinacocyte, which is a flattened, or we can say they are flattened cells, and the second, they are called as porocytes. They are oval in shape. Okay. Well, we have here another layer, and we call that as Charnodome. We call it as Charnodome. We call it as a Charnodome. Now, Charnodome is actually, this is a gastrolayer. It's also called the gastrolayer. Okay, and this is the inner cellular layer. Inner cellular layer. Okay, and the most important thing that it possesses highly specialized flagellated cells. Flagellated cells, and we call them as choanocytes. Most important, choanocytes. These choanocytes are also called as collar cells. Hope you understand. These choanocytes are also called as the collar cells. Well. Now, this forms the characteristic, the feature, we can say the characteristic cells of this phylum. Cells of this phylum. So, when we say that it's a characteristic feature, or we can say the characteristic cells, it means that these kinds of cells cannot be found in other phyla. Hope you understand. Now, these, as I already told you, that they are diploblastic. Well, I will write them on here that they are diploblastic. These are the diploblastic animals. They have only two kinds of layers. Diploblastic animals. I will simply write down it here. It means that they have only two types of the germ layers, or we can say that they have two types of the embryonic cell layers. That's ectodome and anodome. Now, when I say that, it means there is no third layer, there is no mesodome, but there is a kind of, we can say that, a kind of a gelatinous or cream-like or gel-like substance in between these two, that is spinacoderm and the charnodome, and we call them that as mesophyll. We call that as mesophyll layer. Okay, and it's also known as mesenchyma. Mesenchyma. So, for your kind information, it's non-cellular. It is not made up of cells. It's not differentiated into mesoderm. If it would have been differentiated into the mesoderm, then there would be no diploblastic character. There would have been triploblastic character. Okay, so this layer is found in between pinacoderm and charnodome. Because this charnodome is actually, this is the inner layer, and this pinacoderm is actually, this is outer layer, and in between the region, this region is occupied by the mesophyll, or we can say the mesenchyma. Okay, well, this mesenchyma consists of it consists of spongin fibers. Okay, and other numerous spicules. I will show you. They are like the shape of this. Okay, Same kind of shapes they have. Spicules. And it also contains amobasides. Amobasides. And why they are called amobasides? Because they are ammoba-like. They are ammoba-like. That's why they are called amobasides. And these amobasides are found in this mesophyll, or we can say the mesenchyma, that lies in between the pinacoderm and the gynoderm. Okay? We will discuss their role ahead. Now, coming to the another group of cells, we call them as archaeocytes. Archaeocytes. So these archaeocytes, they are actually modified amobasites. They are actually the modified amobasites. Okay, they may be converted into other types of cells, and hence called the undifferentiated totipotent cells. So they are undifferentiated totipotent cells. Well, this was about the archaeocytes, and this all was about the mesenchyme, or we can say the mesophyll. Okay. Now, they have the central body. They have the central body, or we can say, sorry, the central cavity. And that central cavity is called, that's called the sponge seal. Sponge seal. Or paragastric cavity. Paragastric cavity. Okay, this is just like this. This is, for example, the porifera, and this cavity, this is called the sponge seal. Okay, I will show you a diagram, and through diametrically through that diagram, I will revise you what we have studied or what we have learned. Okay. Now, the sponge seal, this cavity, this is actually lined by who? Lined by characteristic cells, that is gynocytes, or we can say the collar cells. Okay, and some canals over there. So canals can also be found that are in contact with these surroundings. Now there is a canal system, and that will be discussed on the next page. There is a canal system. Okay, and we call that as canal system. Okay, so we call that as a water canal system. Water canal system. This is called the water canal system. In other words, we can say also it as a water transport system. So I will write down it here. Transport system. It's important to say water transport system. Okay. So the continuous water current flowing through the canal system is very important for the life of a sponge. It brings in food and oxygen. This water supply, or we can say the canal system, this brings in food, oxygen, and it brings in these things. These things are brought in. I will simply write down as in. These things are brought in into the cavity, or we can say the spongocoel. And the same canal system or the same water system takes carbon dioxide and nitrogenous wastes. It's like ammonia, most important, and reproductive bodies. Reproductive bodies out. Okay, out. So these things are brought in, and these things are brought out. And this happens. These things are brought about by the brought about by the water canal system or the transport system. Well, now we can say, if to summarize this system here, so we can say that this helps in nutrition, similarly respiration, and excretion, and reproduction. So this water canal helps in these processes. So this is about the water canal system and its functioning. As we know, that body is in the form of a cylinder type, this shape, and inside this body cavity, inside it lies the spongocoel, which is actually the large cavity. Now the spongocoel opens out by the narrow opening. We will discuss in that narrow opening that we call it as osculum. The spongocoel is connected with the surroundings, and we call it as the single opening. This is called as osculum. Okay, so this pathway, this helps in the respiratory exchange. This helps in the excretion, or we can say the removal of the wastes. Okay, so this helps with that. Now what? Now let's talking about the another feature here, and that is skeleton. Skeleton. Well, all the sponges, almost all sponges, we can say, they possess an internal skeleton. They possess an internal skeleton. All sponges, they possess an internal skeleton. And this internal skeleton, it may consist of siliceous, or we can say the calcareous spicules. Hope you understand. Or the sponge fibers. Okay, or the sponge fibers, or maybe both. Well, they may consist of this or the sponge fibers, or maybe both. In which layer? In the mesophyll layer. So these things are found in mesenchyma or the mesophyll layer, simply. Now, talking about talking about those sponge fibers. Now, sponge fibers are actually they are silk-like elastic structures. Sponge fibers are nothing. I'll write down it here. Sponge fibers, they are silk-like elastic fibers. They are silk-like elastic fibers. Okay. And talking about the spicules, and what are these? I told you they are formed of silica and calcium carbonate. Okay, hope you understand. So this forms these sponge fibers or the spicules. These form the skeleton. We can say the internal skeleton, and these are found in the mesophyll layer. So in the same mesophyll layer, we can see the amoeboid cells, and these amoeboid cells, well, what's their function? Amoeboid cells or the amoebocytes, their function is to carry food between the cells. So this was about the skeleton system of the sponges. Now talking about the digestion. So what's the digestion? Now how is the digestion process taking place in the sponges? Well, for the, let me choose another color here. I'll write down as a digestion. So what's digestion? The digestion in case of sponges, it is intracellular. Hope you understand what is intracellular. As the term signifies, intracellular. So this digestion, this process that takes place inside the cells, that's why they are called as intracellular. Actually, the sponges are holozoic in nature. They are holozoic in nature. We have discussed it before. They are holozoic in nature. That is, they take solid food as a nutrition, in the form of nutrition, and that's why the digestion takes place inside the cells, and that's why we call it as they have the internal digestion. Okay, and this digestion actually occurs in case of food vacuoles in protozoans, just like as in protozoans, inside food vacuoles, just in case of protozoans. Okay, now talking about another important character, that's circulation. Well, the distribution of the food from the ingesting cells to the others, which I told you before, that is distribution or the circulation of food is actually, so this is actually carried out by amoebocytes. This is actually carried out by amoebocytes, or we can say the amoeba cells. Amoeboid cells, sorry. Now, these amoebocytes, and where are they found? They are found in mesophyll layer, mesophyll layer. And talking about another important character, and that is respiration. So how does the respiration take place in case of sponges? This is another important factor. And now the respiration, actually this takes place, or we can say the exchange of the gases takes place or occurs by diffusion. It occurs actually by diffusion, and this process is based on the concentration of the of any substance. Okay, and this diffusion in case of the sponges, it occurs through the plasma membrane or the plasma lemma. We call it many names, okay, as in case of protozoa. So this characteristic, the digestion characteristic and the respiration characteristic, they are they share these two characteristics in common with the protozoans. Now talking about the excretion. Well, excretion, this is actually the removal of the metabolic wastes. You know the definition now. The removal of the metabolic wastes also occurs By the diffusion through the plasma membranes. Same here, through diffusion, through plasma membranes, through our plasma membrane or the plasma level. So this is the acceleration. Now, now the most important here characteristic, and that is reproduction. Reproduction. Well, the reproduction, first of all, let me tell you that they are hermaphrodites. Hermaphrodites. It means sexes are not separate. Sexes are not separate. Okay? And they reproduce both sexually and asexually. Through both the processes they reproduce. Okay? Well, talking about the asexual reproduction. Asexual reproduction asexual reproduction. The asexual reproduction in case of the sponges takes place through budding. Wait a second. Budding and fragmentation. Fragmentation. Okay. Now, what happens? Talking about spongilla-like sponges, or we can say the poriferans, they are actually, which are actually the freshwater sponges. Now, the freshwater sponges, what they do during unfavorable conditions, they produce buds. But during unfavorable conditions, and we call them as gammules. Keep in mind, please. And we call them as gammules. And these gammules remain dormant till favorable conditions. They remain dormant. And they remain dormant till favorable conditions. Okay. Now talking about the sexual reproduction. Sexual reproduction. During sexual reproduction, some amoeboid cells, some amoebocytes, they get differentiated and they form eggs. Okay, they form egg and maybe egg or sperm. Okay, amoebocytes. That's why I told you that the amoebocytes, they get modified and they form archaeocytes. Well, these archaeocytes, they have the character to form. They have the character of totipotency. They can convert. They can form another cell. So that's why these amoebocytes ultimately they get converted into either eggs or sperms. Okay, they can form an egg or sperm. They can convert into this. So if you after that, what happens then? After that, fertilization takes place, and that fertilization is internal. It's not external, it's internal. Okay. And the development of larva can be seen. You can see larva formation, and which is actually indirect. After the fertilization, what happens? That zygote, this zygote develops into larva. The zygote develops into a larva. Okay, and these larva, they possess flagella. They are ciliated and are free floaters, or we can say the free swimmers. Okay, so these are free swimmers. Well, there are two types of the larva. Two types of the larva can be seen. Let me choose another color here. Well, I will choose red. One's called the amphiblastula. Amphiblastula. And another kind is called the parenchyma. Just for the sake of instance, I will take this amphiblastula can be seen in case of cycon, which is actually also a sponge. And in case of parenchyma, this can be found in Leucosolinia. Hope you understand. So in these two species, they have their respective larva. Now, all sponges, they show great power of the regeneration. They show great power of regeneration. Okay, they have great power of regeneration. And so let me tell you that many sponges, they are harmful. Just like, for example, there is a sponge, and that sponge is called the Chiliona. Keep in mind this point, Chiliona. This is actually the sponge, and it is harmful. And it is harmful to the oyster industry. So actually this is also known as boring sponge. This is not actually that boring that we understand that this guy is boring. This is actually boring sponge. It means that it bores this oyster. When these oysters are, they are like this shape. I will try to make it best. This is found usually in sand. So what it does, it bores this sponge, and that's why. And this is why this is harmful to the industry. Okay, it bores this sponge. Now, and this oyster, as you know, that this contains an oyster, which is a very a kind of a very costly thing, or we can say the resource in this world. Now, talking about some important examples of the sponges. Hope you understand this, because these important examples are important for you. Some common sponges are Leucosolinia, Spongilla, which is actually the freshwater sponge. Okay, this is actually the freshwater sponge. And we have cycon, and after that comes Euspongia. Okay, this Euspongia is also known as bath sponge. Maybe this question can be asked in a NEET, so you should keep in mind. And another important is called as Eupalactilla. Eupalactilla. Okay, this is called as, why I told you this, because this is called Venus flower basket. You will look, if you will see that it really looks like a flower basket. Okay, and it's called Eupalactilla. Okay. Now, what happens? This Eupalactilla is actually, this is used as a costly marriage gift. Costly marriage gift in Japan. When when it gets dried, it forms a costly marriage gift in Japan. Okay, and there it signifies it's a symbol of union till death. Union till death. This gift signifies union till death. Okay, and a few other examples I have here. We have also a Caliona, which I discussed it with you. It's called the boring sponge. It's called the boring sponge, and it's also known as by other name. It's called as sulfur sponge. Okay, sulfur sponge. And another sponge that's called as chalina. Wait a second, got stopper. And another here, it's called as chalina. This is also a sponge, and it's called a dead man finger sponge. Dead man finger sponge. Please keep the names of at least 3 sponges in mind, at least 3, and that's important for you. Well, the last example, and that's called the Proterion. Proterion. It's called as Neptune's cup. Neptune's cup. They exactly look like a cup-like structure, and that's why they have their special names also. So, this was all about the porifera, and let me choose another page here. And if you will see the poriferans, you don't have to remember the classification of the poriferans, but still I will give you. For example, it has been divided into 3 more classes, 3 more important, or we can say the major classes. Class first, and in this class we call them as, this class is known as Calcarea. Okay, Calcarea. And class second, that's called as Demospongia. And this Calcarea, this includes Leucosolia. Okay, and the Demospongia, this is actually the largest class of the poriferans. Largest. Okay, and this includes spongula like sponges. And we have another called the Euspongia. Hope you understand. And the class third, that's called as Hexaactinellida. I'm sure. So this is hexa, or we can say the Hexaactinellida. Okay, well, you can call it, you can pronounce it as, but you have to keep in mind. Okay, they have, and this group includes Euplectilla, and they are called as glass sponges. Okay, they are called as glass sponges with siliceous spicules. So this was all about in the group, the phylum, and the phylum Porifera, and hope you understood the topic. But before concluding, I'm going to show you here the image, or we can say the anatomy of the most important, or we can see the most primitive sponge, and we call that as Solenia. Here we have. So if here you will see that this opening here, this opening is called as osculum. Okay, and these two things, these just like horn-shaped things, they are called as oscular spicules. Oscular spicules. Okay, these two yellow color. Now this entire cavity here, this white entire cavity inside this is called as spongocoel, and here you will find out its name. This is called as spongocoel, or we can say the central cavity. Well, the central cavity, this is actually the trivial name, but the scientific name for this is spongocoel. And here you will see this outer layer, this outer body. It consists of these purple shaped, these pores, or we can say the cells like, well, these are not cells, these are actually the pores. And that's why these pores are created by porocytes. These are actually created by porocytes. Here you can see that these are actually porocytes. So also if you will find out here that porifera are also known as pore-bearing animals. Pore-bearing animals, because they have pores all over the body. Now coming to the another here, these are the pores, and coming to this is outer layer. This is called the panagoderm. This outer layer, this outer layer, this is called the panagoderm. Or we can see the dermal layer, this outer layer, which you can see over here, this entire layer. Now talking about the middle layer here, well, this is called the mesenchyma, or we can say that as a mesophyll. This is called the mesenchyma, or this mesophyll. This layer here, this yellowish color, and this contains this mesophyll layer. They contain these yellow like stars here. Okay, you can see it here. Well, let me zoom this, then you will be able to. Well, this is here, this is yellow like these things here, these yellow like structures here, they are actually called as spicules. Okay, and they are found embedded in this mesophyll, or we can say the mesenchyma. And the inner layer, they consist of these jar-shaped things. They are actually this layer forms the choanocytes, choanoderm, and these cells are called the choanocytes. Well, these are flagellated cells. They have the flagella. Okay, and these are called the choanocytes. And the most important thing here, you will see these bluish things here in the mesophyll layer, or we can say the mesenchyma. These bluish things, they are called as amoebocytes. Okay, so these are the amoebocytes which get differentiated and which form the sperm cells, or which form the egg cells. Okay, so this was all about the anatomy of the sponges, and I hope you understood the topic very well. And getting back to the topic, you will find out that what we have discussed here. So this was totally about the phylum Porifera, and I hope you understood the topic very well. And if you really enjoyed the topic, don't forget to hit the like button below and subscribe my channel, and for further updates. So the next topic will be on the phylum Cnidaria, and till then, have a nice day. Goodbye.